Friends, today we giving you information about most dangerous virus in the world. Coronavirus pandemic. First of all, subscribe my channel for interesting and informative videos. So let's start. Coronavirus disease 2019 COVID-19. Main article: Coronavirus disease 2019 characteristics of patients who have been infected with SARS-CoV-2, MERS-CoV, and SARS-CoV-VTE. In December 2019, a pneumonia outbreak was reported in Wuhan, China. On the 31st of December 2019, the outbreak was traced to a novel strain of coronavirus, which was given the interim name 2019 nCoV by the World Health Organization (WHO), later renamed SARS-CoV-2 by the International Committee on Taxonomy of Viruses. Some researchers have suggested that the Huanan seafood market may not be the original source of viral transmission to humans. As of the 14th of March 2020, there have been at least 5,429 confirmed deaths and more than 145,369 confirmed cases in the coronavirus pneumonia pandemic. The Wuhan strain has been identified as a new strain of betacoronavirus from Group 2b with approximately 70% genetic similarity to the SARS-CoV. The virus has a 96% similarity to a bat coronavirus, so it is widely suspected to originate from bats as well. The pandemic has resulted in serious restrictions over travel. This article is about the group of viruses. For the virus strain causing the 2019-20 coronavirus pandemic, see severe acute respiratory syndrome coronavirus 2. For the disease caused by the virus, see Coronavirus disease 2019. Orthocorona virini. Coronaviruses. Electron micrograph of avian infectious bronchitis virus 2019 nCoV CDC 23312 without background. Illustration of the morphology of coronaviruses. The club-shaped viral spike peplomers, colored red, create the look of a corona surrounding the virion, when viewed electron microscopically. Virus classificationi. Unranked virus. Realm: Riboviria. Phylum: Incerticides. Order: Nitivirales. Family: Coronaviridae. Subfamily: Orthocoronavirini. Genera: Alpha coronavirus, Beta coronavirus, Gamma coronavirus, Delta coronavirus. Synonyms: Coronavirini. Coronaviruses are a group of related viruses that cause diseases in mammals and birds. In humans, coronaviruses cause respiratory tract infections that are typically mild, such as some cases of the common cold, among other possible causes, predominantly rhinoviruses, though rarer forms can be lethal, such as SARS, MERS, and COVID-19. Symptoms vary in other species. In chickens, they cause an upper respiratory tract disease, while in cows and pigs they cause diarrhea. There are yet to be vaccines or antiviral drugs to prevent or treat human coronavirus infections. Coronaviruses constitute the subfamily Orthocoronavirini, in the family Coronaviridae, order Nitivirales, and realm Riboviria. They are enveloped viruses with a positive sense single-stranded RNA genome and a nucleocapsid of helical symmetry. The genome size of coronaviruses ranges from approximately 27 to 34 kilobases, the largest among known RNA viruses. The name coronavirus is derived from the Latin corona, meaning crown or halo, which refers to the characteristic appearance reminiscent of a crown or a solar corona around the virions, virus particles, when viewed under two-dimensional transmission electron microscopy, due to the surface covering in club-shaped protein spikes. Discovery. Coronaviruses were first discovered in the 1960s. The earliest ones discovered were infectious bronchitis virus in chickens and two viruses from the nasal cavities of human patients with the common cold that were subsequently named human coronavirus 229E and human coronavirus OC43. Other members of this family have since been identified, including SARS-CoV in 2003, HCOV-NL63 in 2004, HKU1 in 2005, MERS-CoV in 2012, and SARS-CoV-2, formerly known as 2019 nCoV, in 2019. Most of these have involved serious respiratory tract infections. Etymology. The name coronavirus is derived from Latin corona, meaning crown or wreath, itself a borrowing from Greek koron, garland, wreath. The name refers to the characteristic appearance of virions, the infective form of the virus by electron microscopy, which have a fringe of large, bulbous surface projections creating an image reminiscent of a crown or of a solar corona. This morphology is created by the viral spike peplomers, which are proteins on the surface of the virus. Morphology. Coronaviruses are large pleomorphic spherical particles with bulbous surface projections. 10. The diameter of the virus particles is around 120 nanometers. The envelope of the virus in electron micrographs appears as a distinct pair of electron-dense shells. The viral envelope consists of a lipid bilayer where the membrane M, envelope E, and spike S structural proteins are anchored. A subset of coronaviruses, specifically the members of betacoronavirus subgroup A, also have a shorter spike-like surface protein called hemagglutinin esterase (He). Inside the envelope, there is the nucleocapsid, which is formed from multiple copies of the nucleocapsid N protein, which are bound to the positive sense single-stranded RNA genome in a continuous beads on a string-type conformation. The genome size for coronaviruses ranges from approximately 27 to 34 kilobases. The lipid bilayer envelope, membrane proteins, and nucleocapsid protect the virus when it is outside the host cell. Replication. Infection begins when the virus enters the host organism and the spike protein attaches to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor-attached spike protein. Depending on the host cell protease available, cleavage and activation allows cell entry through endocytosis or direct fusion of the viral envelope with the host membrane. On entry into the host cell, the virus particle is uncoated, and its genome enters the cell cytoplasm. The coronavirus RNA genome has a 5' methylated cap and a 3' polyadenylated tail, which allows the RNA to attach to the host cell's ribosome for translation. The host ribosome translates the initial overlapping open reading frame of the virus genome and forms a long polyprotein. The polyprotein has its own proteases which cleave the polyprotein into multiple nonstructural proteins. A number of the nonstructural proteins coalesce to form a multi-protein replicase transcriptase complex (RTC). The main replicase transcriptase protein is the RNA-dependent RNA polymerase (RDRP). It is directly involved in the replication and transcription of RNA from an RNA strand. The other nonstructural proteins in the complex assist in the replication and transcription process. The exoribonuclease non-structural protein, for instance, provides extra fidelity to replication by providing a proofreading function which the RNA-dependent RNA polymerase lacks. One of the main functions of the complex is to replicate the viral genome. RDRP directly mediates the synthesis of negative-sense genomic RNA from the positive-sense genomic RNA. This is followed by the replication of positive-sense genomic RNA from the negative-sense genomic RNA. 19. The other important function of the complex is to transcribe the viral genome. RDRP directly mediates the synthesis of negative-sense subgenomic RNA molecules from the positive-sense genomic RNA. This is followed by the transcription of these negative-sense subgenomic RNA molecules to their corresponding positive-sense mRNAs. The replicated positive-sense genomic RNA becomes the genome of the progeny viruses. The mRNAs are gene transcripts of the last third of the virus genome after the initial overlapping reading frame. These mRNAs are translated by the host's ribosomes into the structural proteins and a number of accessory proteins. RNA translation occurs inside the endoplasmic reticulum. The viral structural proteins S, E, and M move along the secretory pathway into the Golgi intermediate compartment. There, the M proteins direct most protein-protein interactions required for assembly of viruses following its binding to the nucleocapsid. Progeny viruses are then released from the host cell by exocytosis through secretory vesicles. Transmission. Human-to-human transmission of coronaviruses is primarily thought to occur among close contacts via respiratory droplets generated by sneezing and coughing. The interaction of the coronavirus spike protein with its complement host cell receptor is central in determining the tissue tropism, infectivity, and species range of the virus. The SARS coronavirus, for example, infects human cells by attaching to the angiotensin-converting enzyme 2 (ACE2) receptor. Taxonomy. The scientific name for coronavirus is Orthocorona virinae or coronavirinae. 2, 3, 4 Coronavirus belongs to the family of Coronaviridae. Genus, Alpha coronavirus. For a more detailed list of members, see Coronaviridae. Species, human coronavirus 229E, human coronavirus NL63, Miniopterus bat coronavirus 1, Miniopterus bat coronavirus HKU8, porcine epidemic diarrhea virus, Rhinolophus bat coronavirus HKU2, Scotophilus bat coronavirus 512, genus Beta coronavirus, type species, murine coronavirus species, Beta coronavirus 1, human coronavirus HKU1, murine coronavirus, Pipistrellus bat coronavirus HKU5, Rausetus bat coronavirus HKU9, severe acute respiratory syndrome related coronavirus, severe acute respiratory syndrome coronavirus 2, Tylonicteris bat coronavirus HKU4, Middle East respiratory syndrome related coronavirus, human coronavirus OC43, hedgehog coronavirus 1, ARI-CoV, genus Gamma coronavirus, type species, infectious bronchitis virus species, beluga whale coronavirus SW1, infectious bronchitis virus genus Delta coronavirus, type species, bulbul coronavirus HKU11 species, bulbul coronavirus HKU11, porcine coronavirus HKU15. Human coronaviruses vary significantly in risk factor. Some can kill more than 30% of those infected, such as MERS-CoV, and some are relatively harmless, such as the common cold. 19 Coronaviruses cause colds with major symptoms, such as fever and sore throat from swollen adenoids, primarily in the winter and early spring seasons. 37 Coronaviruses can cause pneumonia, either direct viral pneumonia or a secondary bacterial pneumonia, and may cause bronchitis, either direct viral bronchitis or a secondary bacterial bronchitis. 38 The much-publicized human coronavirus discovered in 2003, SARS-CoV, which causes severe acute respiratory syndrome (SARS), has a unique pathogenesis because it causes both upper and lower respiratory tract infections. 38. 7 strains of human coronaviruses are known. Human coronavirus 229E HCOV 229E, human coronavirus OC43 HCOV OC43, severe acute respiratory syndrome coronavirus SARS-CoV, human coronavirus NL63 HCOV NL63, New Haven coronavirus, human coronavirus HKU1, Middle East respiratory syndrome related coronavirus MERS SARS-CoV, previously known as Novel Coronavirus 2012, and HCOV-EMC, Severe Acute Respiratory Syndrome Coronavirus 2, SARS-CoV-2, previously known as 2019 nCoV or Novel Coronavirus 2019. The coronaviruses HCOV-229E, NL63, OC43, and HKU1 continually circulate in the human population and cause respiratory infections in adults and children worldwide. Severe Acute Respiratory Syndrome (SARS). Main article: Severe Acute Respiratory Syndrome in 2003, following the outbreak of Severe Acute Respiratory Syndrome (SARS), which had begun the prior year in Asia, and secondary cases elsewhere in the world. The World Health Organization (WHO) issued a press release stating that a novel coronavirus identified by a number of laboratories was the causative agent for SARS. The virus was officially named the SARS coronavirus (SARS-CoV). Over 8,000 people were infected, about 10% of whom died. 25. Middle East Respiratory Syndrome (MERS). Main article: Middle East Respiratory Syndrome. In September 2012, a new type of coronavirus was identified, initially called Novel Coronavirus 2012, and now officially named Middle East Respiratory Syndrome Coronavirus (MERS-CoV). 46. The World Health Organization issued a global alert soon after. 47. The WHO update on 28 September 2012 stated that the virus did not seem to pass easily from person to person. 48. However, on 12 May 2013, a case of human-to-human transmission in France was confirmed by the French Ministry of Social Affairs and Health. 49. In addition, cases of human-to-human transmission were reported by the Ministry of Health in Tunisia. 2 confirmed cases involved people who seemed to have caught the disease from their late father, who became ill after a visit to Qatar and Saudi Arabia. Despite this, it appears that the virus had trouble spreading from human to human, as most individuals who are infected do not transmit the virus. 50. By 30 October 2013, there were 124 cases and 52 deaths in Saudi Arabia. 51. After the Dutch Erasmus Medical Center sequenced the virus was given a new name, Human Coronavirus Erasmus Medical Center (HCOV-EMC). The final name for the virus is Middle East Respiratory Syndrome Coronavirus (MERS-CoV). In May 2014, the only 2 United States cases of MERS-CoV infection were recorded, both occurring in healthcare workers who worked in Saudi Arabia and then traveled to the U.S. One was treated in Indiana and one in Florida. Both of these individuals were hospitalized temporarily and then discharged. 52. In May 2015, an outbreak of MERS-CoV occurred in the Republic of Korea, when a man who had traveled to the Middle East visited 4 hospitals in the Seoul area to treat his illness. This caused one of the largest outbreaks of MERS-CoV outside the Middle East. 53. As of December 2019, 2,468 cases of MERS-CoV infection had been confirmed by laboratory tests, 851 of which were fatal, a mortality rate of approximately 34.5%. Diseases caused. Coronaviruses primarily infect the upper respiratory and gastrointestinal tract of mammals and birds. They also cause a range of diseases in farm animals and domesticated pets, some of which can be serious and are a threat to the farming industry. In chickens, the infectious bronchitis virus (IBV), a coronavirus, targets not only the respiratory tract but also the urogenital tract. The virus can spread to different organs throughout the chicken. Economically significant coronaviruses of farm animals include porcine coronavirus, transmissible gastroenteritis coronavirus (TGE), and bovine coronavirus, which both result in diarrhea in young animals. Feline coronavirus, 2 forms. Feline enteric coronavirus is a pathogen of minor clinical significance, but spontaneous mutation of this virus can result in feline infectious peritonitis (FIP), a disease associated with high mortality. Similarly, there are 2 types of coronavirus that infect ferrets. Ferret enteric coronavirus causes a gastrointestinal syndrome known as epizootic catarrhal enteritis (ECE), and a more lethal systemic version of the virus, like FIP in cats, known as ferret systemic coronavirus (FSC). There are 2 types of canine coronavirus (CCOV), one that causes mild gastrointestinal disease and one that has been found to cause respiratory disease. Mouse hepatitis virus (MHV) is a coronavirus that causes an epidemic murine illness with high mortality, especially among colonies of laboratory mice. Sialoda cryoadenitis virus (SDAV) is highly infectious coronavirus of laboratory rats, which can be transmitted between individuals by direct contact and indirectly by aerosol. Acute infections have high morbidity and tropism for the salivary, lacrimal and Harderian glands. A HKU2-related bat coronavirus called swine acute diarrhea syndrome coronavirus (SADS-CoV) causes diarrhea in pigs. Prior to the discovery of SARS-CoV, MHV had been the best-studied coronavirus both in vivo and in vitro as well as at the molecular level. Some strains of MHV cause a progressive demyelinating encephalitis in mice, which has been used as a murine model for multiple sclerosis. Significant research efforts have been focused on elucidating the viral pathogenesis of these animal coronaviruses, especially by virologists interested in veterinary and zoonotic diseases. Thanks for watching till end. Be careful friends and be safe always. Be happy friends. Take care yourself and your loved ones. Please subscribe my channel for more informative videos.